Thanks everyone for coming, joining in for my talk today, and thanks for joining in for the symposium stemming from our Manakin Research Coordination Network. Today what I'm going to try to highlight for you is some of the efforts stemming from this network aimed at understanding patterns of genome evolution in this group and relating those patterns to sexual selection. As many of you know, there's been kind of amazing changes in the worlds of genome biology over the last 15 or so years. In particular, the sort of proliferation of genomes across the tree of life, and nowhere is that more true than in birds. In manakins, we're doing pretty well as well, and largely as a result of this RCN, over the last 5 years, we moved from having a single genome in the group to having a nice sampling of lineages. We're up to 6 genomes now. But what's even more exciting than that, I'd say, is that beyond genome assemblies themselves, the genomic resources and the people associated with the development of genome resources have really grown. And so there's a large community of researchers working on these exciting birds and bringing genomic resources to bear to answer all kinds of questions in this group. And hopefully you've heard about some of them today, but a lot of exciting stuff should be coming forward as these tools continue to develop. And so today in this talk, my goal is to tell you really about the work that my lab has been closely involved with. First, starting with a broad overview of the genomes we've sequenced to date and how they're informing our understanding of sexual selection from a genomic perspective. Second, diving into a single display component in detail to look at kind of the underlying muscle physiology from a transcriptomics perspective. The third project my lab has been really involved in gets at the brain and relationship with behavior. And if you want to hear about, although I'm not going to talk about that stuff today, if you want to hear about that work, I encourage you to tune into this talk from my postdoc, Dr. Perry Bolton. And so to begin with the genomes, I'll emphasize here that this is a large collaborative effort of which I have the honor of being the voice today, but I'll try to highlight throughout this talk the work of a lot of the key people involved. But this is a RCN driven comparative analysis and has involved the work of a lot of people. And as you've almost certainly heard of throughout the day, the exciting thing that draws people to manakins is really this elaboration of courtship displays and all the wild and wacky things that we know they do. Today, though, I want to highlight another part of Manakin biology, and that is an interesting transition that we observed within the group from towards a more frugivorous lifestyle. And so this transition between more insectivorous species and more frugivorous species occurs within the manakins and in association with this enhanced display complexity. And that has led some authors to speculate, in fact, that this transition in diet is actually causal in allowing for the enhanced sexual selection and the increased amount of time that males spend in doing displays in their daily lives. And so the way we're thinking about this is really from a physiological perspective and trying to relate changes in the genome to changes in diverse components of physiology that must evolve in an integrated way in order to support the behavioral novelties that we observe. And so for this first part of the talk, I'll highlight first the genome assemblies and then try to put a genome-wide perspective on signatures of selection. So how does sexual selection impact the genome as a whole? And can we identify key parts of the genomes that are involved in the origin of these novel traits, including this transition in diet? And finally, we'll take some of those and really delve into the nitty-gritty of how they're working mechanistically. And so here I've highlighted our first 5 genomes sequenced, and hopefully you can see that what we've got is a nice sampling across some of the key lineages in manakins. But importantly, and perhaps most importantly for the context of this study, one of the lineages we've sequenced represents this clade called Neopelma, which relative to the rest of manakins has relatively simple displays and relatively less frugivorous diet. And so having this taxon allows us to sort of map changes in the genome onto this important transition point in Manakin, display, and phylogeny. And then we can take all 5 of these genomes and compare them to outgroups to really understand traits that are shared by all Manakins. And so that's highlighted by this figure here that really gets at our sampling strategy, where we have our outgroups, our Manakins as a whole, including the relatively insectivorous and monomorphic Neopelma, and then what I've called here the fancy Manakins. Briefly, these are the approaches we've taken, which have evolved with changing technology and continue to evolve to give us really nice genomes. So first, genome-wide signatures. The first thing we wanted to look at really is to test a simple prediction that strong sexual selection should reduce the effective population size of the Z chromosome, or in other words, diversity on the Z chromosome. And the idea here is first that because the Z chromosome is only present in 3 copies for every 4 copies of autosomes, because females are heterogametic, we expect the Z chromosome to have lower diversity than autosomes. But because of reproductive skew among males, we expect further reduction of diversity in that Z chromosome diversity relative to autosomes as compared to outgroups that don't have strong sexual selection. And so first, if we look at heterozygosity here from our male genomes, that is where we have 2 copies of the Z sequence, so we can look at heterozygosity. We see the expected patterns. There's lower diversity on the Z in purple relative to the autosomes in all of the other colors. We can take these diversity estimates, these variants that we can identify in these species, and use an approach called PSMC to reconstruct historical demography and effective population sizes. And here we're going from more recent in time on the left to further back in time on the right. But the key part here is that the effective population sizes estimated for the Z are substantially lower than the autosomes. And in fact, lower than that neutral expectation of 75%. And we're looking at this now in more detail, looking at population genetic variation, larger sample sizes from some of the key species in this study. So we'll have more to report on that soon. But taken together, our results are pointing towards an important effect of sexual selection on polymorphism of the sex chromosomes. And those effects could be predicted to have consequences for how genes on the sex chromosomes evolve. So Z chromosomes across all species of birds evolve faster than autosomes. And at lower effective population sizes, we'd affect genetic drift to have a larger effect, and therefore further increase the rate of evolution in Manakins relative to outgroups. But that's not what we see, actually. We see the expected acceleration of the Z relative to autosomes in Manakins and the outgroup species that we looked at, but we don't see this enhanced acceleration of the Z chromosome, which suggests that this drift is really not playing a major role in modulating this acceleration of the Z chromosome. To zoom in further, to identify specific targets of sexual selection, or selection in the clade, selection more broadly in the clade, we've looked at both protein coding genes, and that's work that's been done by Robert Driver, as well as conserved non-coding elements that represent putative regulatory elements. And you should have just heard about that from Dr. Morgan Worthlin. And if you want to see that talk, I suggest you go back and look at it for the details there, because I'm not going to present them here. And so I'm presenting these results from selection tests on protein coding genes and kind of that framework of the different physiological domains that I mentioned earlier. And we can see some really striking patterns, including here, selection on genes with known roles in cardiac and muscle systems, including a troponin, adrenoceptor, and desmoglein 2. We also see very compelling signatures of selection on digestive enzymes. So here on the branch leading to the fancy Manakins, we see selection on sucrose isomaltase, as well as fructose biphosphatase, 2 key sugar metabolism genes. And if we zoom back a node in the tree, again, on that node, we see continued selection on sucrase, the sucrase gene, as well as this kind of wacky finding where we find selection on lactase. And so lactase is well known as in mammalian systems for the role of this enzyme in breaking down milk. And so when we saw this result, we were sort of confused, but it turns out the full name of lactase is lactase fluorescein, and this second component may provide some clues as to what it's doing in these birds. And so fluorescein is a plant compound, and so perhaps the role of this lactase enzyme in in mannequins here also has to do with the breakdown and digestion of plant compounds derived from the fruits that they eat. But that is ongoing work that we're trying to understand better, but still pretty exciting, I think. We also see interesting things going on in sensory systems. In terms of the visual system, we see selection and that branch leading to the fancy mannequins on the short wavelength sensitive opsin. And in this part of the tree, we also see selection on taste receptors, which could be involved in, again, the dietary shift. For more on the short wave sensitive opsins, I'll point you to a talk from Robert Driver, again, part of this RCN and part of my lab, and so check it out. But again, if we zoom out or back one more node in the Manakin tree, we see selection on another opsin, OPN4RM, that Robert will tell you more about, and again, continued selection on a portion of this T1R3 taste receptor. And so to take these results a little bit further, we've been diving into functional testing of these, including in situ hybridizations for the genes associated with these putative regulatory elements that Morgan would have told you about already. But we've also dove in a little deeper to the taste receptors to see, do these really respond to the sugars found in fruits? And to do this, this has been a lot of work from Maud Baldwin and Yaska Toda. They have sort of developed these approaches for a really cool study from 2014 where they described sweet taste perception in hummingbirds. And the quick punchline there is that, you know, most birds don't have a sweet taste receptor. And in the context of nectarivory, hummingbirds have convergently evolved sweet taste perception using this T1R1, T1R3 taste receptor pair that form a dimer. And we wanted to really see if the same or kind of thing was happening in mannequins in the context of frugivory. And so in a cellular assay, these receptor pairs have been tested in ant birds and mannequins. And so this figure on the left kind of highlights the basic approach where in a cellular context, the receptors are tested for responsiveness to sugars as well as amino acids. And you can see that the barred antshrake, mostly insectivorous species, does not respond to sugar stimulation. And neither does the rusty margin flycatcher, but both Manakin species receptors that are tested do respond to sugars. And so this is super cool because it both shows another convergent origin of sweet taste perception in birds, in this case, in the context of frugivory as opposed to nectarivory. And again, this Neopelma taxon is kind of interesting because it puts a timeline on when this happens, and it sort of supports a scenario in where there was this shift in diet early in the Manakin regulation and even before the real elaboration of courtship displays has occurred. And so far here, I've shown you effects of natural and sexual selection on these diverse physiological systems. I haven't touched too much on neural control systems and endocrine systems, perhaps, and there are other talks in this symposium, like some of those that I've mentioned, that I'll get into that in a lot more detail. But what I wanted to do next really is focus on one of these in more detail, and that is the musculoskeletal system. So in this part of the talk, what I wanted to do is to delve into detail rather than looking at sort of the elaboration of courtship displays generally and sexual selection generally. I wanted to move in to talk sort of into the detailed dissection of a single and specific display, this rapid wing display that's found in a number of Manakin species. And here what we've done is we've taken a comparative transcriptomic approach to look at changes in gene expression associated with rapid wing displays. And so this builds on work from the Matt Fuchsjager lab and is a collaborative project that is stemmed from the RCN. And so although I'm presenting this work, I'll really highlight that this work comes from Matt's lab and has involved really hard work from the two leads on this project, James Pease and Robert Driver. Here I wanted to present you with the phylogenetic context for this study, which is similar to previous, but slightly different players involved. Here we have Myanectes as an outgroup relative to a set of 6 manakins that we sequenced for transcriptome data and 1 Pipistrellocauda that we used its reference genome for these analyses. I also wanted to place these taxa in the context of that display elaboration that we've been talking about, where you can, and this using an actual quantification from this study by Lindsay and colleagues from 2015. And so what you can see here is that the outgroup Myanectes has a relatively simple display, whereas across manakins, you have relatively high display complexity, but also you can see variability. For example, Xenopipo has a relatively low display complexity, and then nested within a set of manakins that have high display complexity, Pseudopipra has low display complexity, more similar to some of the outgroups and some of their closest relatives. And within this context of variation in display complexity in manakins, we have the specific display that we're interested in that is this rapid wing movement, a rapid wing display. In Manicus vitellinus, this is the sort of well-known wing snap where the wing is lifted up at a remarkable speed to produce acoustic sonation, and kind of a neat, very neat example of non-vocal communication, and a similar kind of rapid wing movement in the Ceratopipra manakins. And because of the previous work for Matt and his colleagues, we actually know a little bit about the underlying physiology that supports these cool displays. And so for one thing, the muscles, wing muscles in these manakins with these rapid wing displays have enhanced androgen receptor expression that support these displays. But what muscles might be critical to generating these displays? Matt and his colleagues have also shown that the scapulohumerales caudalis, this SH muscle, which is the muscle that's involved in the wing uplift, shows this really high enhanced contractility, and that other muscles of the wing do not. And so it seems like changes in this SH muscle in particular were a key component of this evolutionary novelty. And so across those taxa that I mentioned, what we've sampled is gene expression in both the SH and the PEC for 3 samples of each of those Manakin species and the Myanectes outgroup. And doing so has allowed us to study changes in gene expression in the 2 muscles across phylogeny to look at the unique changes in both muscles associated with wing displays, and in particular, those changes in SH that are tightly correlated with this enhanced muscle contractility. To provide a brief and comprehensive kind of overview, big picture overview of the data here, I've just laid out a PCA for you. It's important to mention here that all of these samples are field caught. These are males displaying on territories. And for any field study, when you're studying gene expression, it's really nice when you see samples actually clustered by species, because there's a lot of things that can impact gene expression, and pulling out species-specific patterns of gene expression across 2 muscles is really satisfying and reassuring. But even more cool is that we can also pick up some phylogenetic kinds of signatures. And so if you look at PC2, you can see low on PC2, we have some of our outgroup Myanectes, as well as as well as Xenopipo, another one of these species with a simple display. And so along PC2, we kind of have a separation of species that have elaborated courtship displays and those that have relatively simple displays. And this isn't purely phylogenetic, because you can see that Pseudopipra, the taxon that I highlighted earlier that has a simple display, actually groups with these outgroup taxa that have also relatively simple displays. And so that's kind of a neat signature in the data that's perhaps enhanced in the SH relative to the PEC, where you have this really nice tight clustering of those 3 taxa with simple displays in the SH. Also in the SH, we see this clustering of the 3 species that have the rapid wing movement. And so that's also a really exciting sort of overview feature of these transcriptomes in general. And we can look at that another way in terms of hierarchical clustering, where here again, I have the PEC clustering patterns on the left and the SH on the right. And what you can see is that for the PEC, the pattern of hierarchical clustering largely reflects the pattern of phylogenetic patterns of gene expression, whereas the patterns on the right for the SH more closely reflect display behaviors. And so the three species that have the rapid wing displays largely clustered together, which is contrast with their phylogenetic relationships. So here what we've plotted are differential expression volcano plots between the two muscles. And so within each species, we contrasted gene expression in our 3 PEC and our 3 SH samples. And the most striking result of all of this is that our mannequins with greater display complexity show much more differential expression between the two muscles. And that's highlighted here in these bar plots as well that show genes upregulated in SH for each taxon and genes upregulated in the PEC for each taxon. But the scope of the size of these bars is the total number of differentially expressed genes. And you can see for Lepidothrix, Manicus, Pseudopipra, and the Ceratopipra mannequins, we have many more differentially expressed genes. Also, I've highlighted here with the stars, the 3 taxa with the rapid wing movements. But given the huge number of differentially expressed genes in Lepidothrix, this differential expression pattern doesn't really map on to rapid wing displays very tightly. Rather, what we see is that along with display complexity evolution in the mannequins, we see this enhanced differentiation of these two muscles. What kinds of genes do we see differentiating SH and PEC muscles? For Manicus and Ceratopipra, we actually had overlapping gene ontology categories related to actin binding and actin filament sliding. For those two plus Lepidothrix, we actually see changes in energetics, so a little deeper in the tree. These two muscles are differentiating in terms of their cellular energetics. What we really want to do here is chart changes in gene expression in a phylogenetic context. And this is an area that's been in search of methods for some time. And to pull this off, we were able to recruit James Pease from Wake Forest and David De La Cerda to help develop methods and take on these analyses. And so really what we wanted to be able to do is distinguish for each gene, compare models of expression evolution, and ideally testing for different evolutionary scenarios, such as independent gains in association with rapid wing displays in Ceratopipra and Manicus, gain loss kinds of models, as well as identifying genes that show changes along the Manakin phylogeny in terms of building up the architecture required to support gene expression displays. And this has gone really well thanks to their hard work. And so here I'm just highlighting kind of what this looks like. And so here, these are genes where the best fit model supports a change in gene expression kind of at the base of the Manakin. So where the 6 Manakin taxons show a difference relative to the Myanectes outgroups. We can do the same thing for the next node up in the tree. I'm not going to belabor any of the genes or the details here, but just show you that these methods, these phylogenetic comparative methods for looking at gene expression are working really nicely. Where it gets trickier is these more complicated models of evolution and testing these kind of key questions related to changes in specific independent lineages like those that have the rapid wing displays. And so here I wanted to start by showing you an example that we know about from previous studies. We expect higher androgen receptor expression in Manicus and Ceratopipra. And in fact, that's what we see. And so here I displayed 2 bar plots of expression. Each sample gets a bar, each taxon is color coded with the rapid wing movers starred again at the bottom. And so I've shown this as log 2 counts per million and raw counts just to give you different views of the data. And what you can see is the expected result here, that androgen receptor expression is higher in those rapid wing movers. And that we already knew going into this. But on the right here is kind of the really complicated bit where, again, on the left for the SH, on the right for the PEC, we've looked at model fits for different models, different phylogenetic models of evolution. And that ranges from independent lineage-specific models to these convergence models. And what turns out for this androgen receptor gene, the model with the best fit is a convergence model where Manicus and Ceratopipra have independently evolved this elevated gene expression relative to the other species. Now you'll note that this delta log likelihood that's highlighted here is not particularly great. And so this is only marginally better than the null model, but still supports this change in androgen receptor in association with this display. As another example, I wanted to show you this gene supervillain, which again shows changes in Manakin as well as Ceratopipra. But interestingly, in this gene, we really only see this change in SH. So these are the kind of signatures we're interested in to identifying key regulatory changes that are specific to the SH. In this case, we also see this change in Lepidothrix. So this might move that change further back in the phylogeny. And so the model that's the best fit is actually a different model. It's a reversion model where Pseudopipera has lost this change in expression in the SH. Here's just a couple more models that highlight different kinds of genes. So these are structural proteins involved in basically in muscle structure. These are two key acting proteins, titin and nebulin. And again, these two genes here, just showing you the raw counts, these are really highly expressed genes that show elevated expression in both SH, specifically the SH, but in both Manakin and Ceratopipra. So these are really important structural molecules that might play a role in this enhanced contractility of the muscle. And so what I've tried to show you is that we've identified a host of gradual changes in gene expression along with the elaboration of courtship displays. So there's big changes in these muscles, along with the enhanced differentiation of these two muscles. So it's not all the changes it's not just that muscle expression as a whole is changing, but the relationships between the two muscles are changing in the phylogeny across all mannequins, and specifically in those mannequins that have rapid wing displays. Pseudopipra is a particularly interesting taxon, I think, in this analysis, in that in multiple ways it kind of shows reversion-like patterns, reverting to an expression state similar to those mannequins deeper in the tree that don't have complex displays. And so in association with these rapid wing movements, what we've identified so far are changes in gene, and some of which I haven't admittedly had time to talk about. We've identified changes in genes associated with cellular energetics, both muscle strength and speed. And so I wanted to end this talk, I just wanted to revert back to my starting slides where I highlighted the wealth of genomic resources now available across Manakins. And some of this is directly stemming from the RCN, and others is just building upon a long history of work in this group. But as you can see, there are also lots of people involved in this research. In fact, many, many people working on Manakins are not shown here. I've tried to highlight those most tightly associated with the genomic work. But there's a lot of exciting work going on in Manakin biology, in Manakin land, and having these type of genomic resources in a well-defined clade with really interesting behavior and evolutionary history, I think is really setting the stage for a lot of cool stuff to come. So thank you again for joining for this talk and for whatever parts of the symposium you can see. I encourage you to check out the rest of the talks, and thank you very much.